5.1 The cell cycle. There are three main ideas. The cell cycle has four main stages. Cells divide at different rates, and cell size is limited. There are three vocabulary words for this section: cell cycle, mitosis, and cytokinesis. Please follow along as I read this aloud. Connect. Many of life's little chores, such as sweeping and dusting, are quietly satisfying and rather fun. Washing dishes by hand, however, is never fun, which is why some clever person made the dishwasher. This handy invention soaks, washes, and rinses your dishes to a spot-free sanitary sparkle. You unload the dishes and the machine is ready to start the cycle all over again. A cell goes through a cycle too. This cycle of growth, DNA synthesis, and division is essential for an organism to grow and heal. If it goes out of control, abnormal cell growth may occur, resulting in cancer cells like those shown on the previous page. Main idea: the cell cycle has four main stages. Just as all species in life have cycles, from tiny chihuahuas to massive beluga whales, cells also have a cell cycle. The cell cycle is the regular pattern of growth, DNA duplication, and cell division that occurs in eukaryotic cells. Figure 5.1 shows its four main stages: gap 1, synthesis, G2, and mitosis. Those are indicated right here: gap 1, S, or synthesis, G2, or gap 2, and then mitosis, which has several subsections. The stages of the cell cycle get their names from early studies of cell division. Scientists' observations were limited by the microscopes of the time. When a cell was not actively dividing, they could not see activity in it. Thus, they originally divided the cell cycle into two parts: interphase, when the cell appeared to be at rest, and mitosis, when the cell was dividing. Improved techniques and tools later allowed scientists to detect the copying of DNA, which is DNA synthesis, and they changed the description of the cell cycle to include the synthesis stage. Since they still could not see anything happening during the other parts of interphase, scientists named the periods between mitosis and synthesis gap 1 and gap 2. Eventually, scientists learned that during interphase, cells carry out their normal functions and undergo critical growth and preparation for cell division. And we can see interphase indicated on this chart beginning right here and going around to right here. So there are two sections of the cell cycle. There is interphase and there's mitosis. In interphase, we have these three things included: gap 1, synthesis, and gap 2. Gap 1. The first stage of the cell cycle is gap 1, otherwise called G1. During G1, a cell carries out its normal functions. If it is a skeletal muscle cell, it contracts to move joints. If it is an adrenal cell, it secretes hormones such as adrenaline. If it is an intestinal cell, it absorbs nutrients. During G1, cells also increase in size and organelles increase in number. A cell spends most of its time in the G1 stage, although the length of this stage varies by cell type. During G1, the cell must pass a critical checkpoint before it can proceed to the synthesis stage. Just as it would be dangerous for you to run a marathon if you had not slept or eaten for several days, it would also be dangerous for your cells to continue dividing if certain conditions were not met. For instance, most animal cells need enough nutrition, adequate size, and relatively undamaged DNA to divide successfully. They also need specific signals from other cells, telling them whether more cell division is needed. Synthesis, or S stage. The second stage of the cell cycle is the synthesis, S stage. Synthesis means the combining of parts to make a whole. During the S stage, the cell makes a copy of its nuclear DNA. In eukaryotes, DNA is located in the nucleus. During interphase, it is loosely organized and appears grainy in photographs. By the end of the S stage, the cell nucleus contains two complete sets of DNA. And with the S stage, we had spoke previously in first semester about DNA replication. DNA replication, once again, is where the DNA separates and results in two identical strands of DNA. In the cell cycle, that happens in the S phase. Gap 2, or the G2 phase. Gap 2 is the third stage of the cell cycle. During G2, cells continue to carry out their normal functions. and additional growth occurs. Like G1, this stage includes a critical checkpoint. Everything must be in order. Adequate cell size, undamaged DNA, before the cell goes through mitosis and division. Mitosis. Mitosis, or M, the fourth stage of the cell cycle, includes two processes, mitosis and cytokinesis. Mitosis is the division of the cell nucleus and its contents. During mitosis, the nuclear membrane dissolves, the duplicated DNA condenses around proteins and separates, and two new nuclei form. Lastly, cytokinesis is the process that divides the cell cytoplasm. The result is two daughter cells that are genetically identical to the original cell. The stages of the cell cycle and the proteins that control it are similar in all eukaryotes. For example, scientists have demonstrated that some of the molecules that regulate checkpoints in the yeast cell cycle can work in human cells too. Such similarities suggest that eukaryotes share a common ancestry.